Up to this point, our review of the process used in evaluating the biocompatibility of implantable materials and devices covered tests which were developed and are used in many traditional fields and laboratories, such as biomedical engineering, material science and engineering, biology, and biochemistry. All of these tests are in vitro laboratory tests. At this particular point, some approaches which are unique to biomedical engineering applications will be reviewed, and they are animal tests and clinical trials. In lecture 28, we'll review the part of the evaluation process for the biocompatibility of implant materials and devices, which involves animal tests. The important prerequisite before any animal tests are considered is the following. Animals in research and in testing should be used only after successful completion of all preceding tests, which involve the material mechanical properties, other pertinent in vitro experiments, such as the one that use culture cell models, etc. Another unique aspect of this stage of evaluating the biocompatibility of implantable materials and devices is that use of animals in research is regulated by federal laws. And use of laboratory animals is protected by these federal laws in the United States. The history of these laws started with the Animal Welfare Act, which was signed in 1966. There were subsequently various amendments which were passed in 1970, 76, 85, 1990, 2002, 7, and 2008. The Animal Welfare Act is enforced in the United States by the Department of Agriculture, also known as USDA. There is a special unit in the department, the Animal Care Unit, which is part of the Animal and Plant Health Inspection Service. Following pertinent investigation by the federal authorities and determination of violation to these laws may result in criminal penalties, civil penalties, fines, and last but not least, closing down the facilities which conduct research using animals in laboratories at universities, companies, and other such institutions. Biomedical engineers who are interested in implantable materials and devices should be, first and foremost, should be aware of the existence of these federal laws. In addition, biomedical engineers should know the main points of these particular laws. That's what we're going to do briefly now. The Animal Welfare Act of 1966 was the first one to establish policies and standards regarding the handling, the sale, and transport of animals, specifically cats, dogs, non-human primates, rabbits, hamsters, and guinea pigs. This group of animals was mentioned in the Animal Welfare Act of 1966. In addition, providers and or dealers of animals and the laboratories where these animals are used for experiments had to be licensed, and in addition must provide identification of their animals in terms of where these animals came from. This particular requirement was included in order to prevent animal theft and exploitation. Up to that point, there were very sad incidents of pets which were stolen, and people found them in in places where research was conducted, or sometimes there were groups of people who went around towns and collected stray cats and dogs and sold them for research purposes. These particular practices are strongly prohibited by the Animal Welfare Act and all the subsequent amendments. In addition, the Animal Welfare Act, with the various amendments, was expanded and included details regarding the care of animals in commerce, exhibits, teaching, testing, and research. There were variations or amendments, I should say, to the original Welfare Act of 1966, and the amendment, which is very important, is that it included all warm-blooded laboratory animals under the protection of these laws. Cats, dogs, primates, and other mammals were included, but birds, rats, and mice were excluded from the protection of this particular law regarding the use of this type of animals in research. Subsequent amendments expanded and refined the original Animal Welfare Act. Specifically, the amendment of 1985 addressed more specifically the welfare of animals in the laboratories, the testing of animals, animals on exhibit, and captive marine mammals. The United States Congress established an information service. This is known as the Animal Welfare Information Center at the National Agricultural Library. This particular center provides information and resources regarding the care and welfare of animals, and is also providing information and resources for alternatives. Those alternatives are now known as the 3 R's: reduction, refinement, and replacement. Specifically, aiming at reducing the number of animals which are used for tests, even when the purpose is biomedical research, refining the techniques and the appropriate test, and last but not least, developing novel and creative ways to replace the use of animals. And of course, one of those more recent developments was the use of culture cell models. One of the requirements, the foremost requirement of the Animal Welfare Act and all the associate amendments is the priority which is placed on the fact that the animal welfare needs must be met at all times. One of the way that this particular need can be addressed is the establishment of committees known as Institutional Animal Care and Use Committees, abbreviated IACOCs. These committees were established in order to oversee and assure proper animal care and animal welfare at registered institutions. A specific example is the Institutional Animal Care and Use Committee at UTSA. Researchers who want to conduct animal trials at UTSA must first obtain approval of their protocols from the UTSA IACOC. This committee is composed by faculty and staff who have experience with animal-related research, and it's chaired by the UTSA veterinary physician. The submitted application should present detailed description of animal handling before experimentation, during, and at the end of the study. The submitted protocols must follow the federal law requirements for animal trials. Experiments may start only after the UTSA IACOC approves the proposed protocol and not before. Pre-approval of animal-related protocols by institutional IACOCs is required before consideration of an animal research-related grant application, which is submitted to national funding agencies, specifically the National Institutes of Health, NIH. Biomedical engineers who are considering animal tests in the process of determining the biocompatibility of their materials and devices should be aware of the following specific requirements. The animal research regulations identify and specify the protected animal species. For example, pets are included in that category. Farm animals are not. How these animals are procured for the study. For example, just to obtain the stray cats and dogs from the city, which may be a desirable outcome to clean the streets of the city, is not allowed. In fact, animals these days which are used for research are raised in farms which have that purpose only. They're licensed and they're established, and they have established their professional credentials in such a way that if biomedical engineers state in their protocols which are submitted to IACOC or to NIH that I will use these animals from such specific farm, which has now, of course, impeccable professional credentials, this particular aspect is accepted by the appropriate committees. And this is a very important aspect to convince the committees that the particular study is going to start on the right foot and in the right direction. How these animals will be expanded or grown. Sometimes, for example, long studies determining the molecular and these days gene level of various diseases require various generations and multiple generations, let's say, of mice. And this particular kind of study will require not only to obtain the animals from a farm which raises animals for research, but it will require to generate the various generations of these mice at the facilities where the research will be conducted. The laws regarding animal tests cover this particular aspect as well. Therefore, appropriate applications by biomedical engineers must cover in detail and provide all information this particular aspect. Veterinary care of animals upon arrival, before, during, and after any study, and at the end of the study, is required. Animal facilities, for example, the one that we have at UTSA, has a veterinary physician who is in charge of taking care of these particular animals. Definitely, if the animals are procured from a professional animal farm, the farm assures the researchers that these animals are healthy, but in addition, upon arrival, the resident veterinary physician will examine the animals, and he or she and their assistants will take care of the animals before, during, and at the end of the pertinent study. An institution where animal tests are conducted must also provide an appropriate animal facility. The Animal Welfare Act and the pertinent amendments specify and define the regulations pertinent to the animal facilities. These details include how clean and how often and how they are maintained clean, the temperature control of the facility at all times, the durations of lights on and off, because of course the animals are now housed indoors and therefore the lights on and off control the rhythm, biological rhythm in terms of their life's requirements, the dimensions of the animal cages. Bigger animals will require bigger cages. Smaller animals, of course, smaller cages, but even then, the number of animals per cage is specified. Just because they're little mice, one cannot pack a particular cage with mice. So many animals are allowed per cage, and the dimensions of the animal cages are specified pertinent to the size of the animals. The availability of water and food is also a requirement, and it must be described in the protocols of the people who want to conduct this particular research. Of course, if the animals are going to undergo surgery, then the food will not be given to the animals prior to surgery, similarly with what happens with humans undergoing surgical procedures. Availability of water is, though, however, something that must be available to the animals at all times. Special animal requirements are also specified by these laws. For example, dogs, which are not used as much in research these days, but if they are used, the facility should provide a room or space for exercise. These are runners, and the animals, the dogs, must be brought to the runners and allowed to run or move around a couple of times a day. If primates, and those are animals which are not used very much at all these days because there are not too many of them, and in addition, their use in experimental research is very, very strictly controlled. If primates are used, then the psychological well-being of these animals has to be addressed and taken care of as well. In addition to having animal facilities, institutions where research is conducted using animals must also provide information regarding who is running these facilities. The training and credentials of animal care personnel must be provided and approved by the various committees, such as IACOC, committees at NIH, etc. The credentials of veterinary physicians and assistants should also be included and approved. The credentials and experience of surgeons, the ones who are going to conduct the procedures, surgical procedures, to introduce the implantable devices in the bodies of the animals. Last but not least, the credentials and experience of the researchers who are going to conduct the animal trials are also reviewed and must be approved by the appropriate committees. Applications seeking approval of experimental protocols using animal tests are lengthy documents and must include the following information: detailed description of all experimental procedures which include animals, pertinent controls at all stages of the study, detailed surgery procedures, anesthesia which will be used during the surgery, or other tranquilizing drugs, pre and post animal care that will include information who is going to be taking care of these animals, therefore the accredited personnel for the animal facility, what medications will be used, for example, the type, the dose, and the duration of the delivery of such medications. How will the animals be euthanized at the end of the study? What medication will be used, what dose, and what are the potentially adverse effects. The medical treatment of animals when they are sick for reasons are related to the study. I emphasize this particular aspect because if the animals get sick and that particular development is related to the particular study, the implantable material and device, that particular result is included in the documentation of the data from the study. But if, for example, the animals get sick because they get a cold and the study pertains to an implantable device, this is a case of a sickness which is not related to the study, and therefore the animals will be treated by the veterinary physician using, for example, some appropriate medication. This detail, though, should be included in the description of the protocols which are submitted for approval by the appropriate committees. Last but not least, how the animals, the dead animals at the end of the study or during the study, if they die during the study, this information of how those dead animals will be disposed should also be included. In addition to detailed and lengthy applications which must be provided to the appropriate committees in order to obtain approval to conduct animal tests, animal experiments require lengthy and very detailed, meticulous record keeping. And these aspects include all experimental procedures, the daily log of what happened with the particular study, description of the pertinent controls and what the results were regarding the controls versus the animals, which are, of course, the main part of the study. All surgery procedures, pre and after surgery treatment of the animals, statistical analysis of the data. All results of the study must be included in the record keeping for the duration of the particular study. All these records must be kept because they are required sometimes at regular and specific time intervals by the appropriate regulatory agencies, such as the Institutional Animal Care and Use Committees, the IACOCs, national funding agencies such as National Institutes of Health, NIH, and last but not least, by the Food and Drug Administration, FDA. Keep in mind that the FDA evaluates the biocompatibility of medical devices before approving them for legal marketing. Therefore, all this documentation must be collected, maintained, and submitted at the end of the study to the FDA when the researchers are seeking approval of their product in order to get permission to market legally this particular product. Use of animals in experiments and tests has certain critical requirements which engineers in traditional disciplines do not encounter and they are not aware of. As biomaterials, biomedical engineers, we must be aware of these critical requirements. First and foremost, all animals in experimental studies should be treated with humane care. What that involves is that all animal tests and experiments should not expose the animals to unnecessary pain and suffering, and not large numbers of animals should be used. In fact, large numbers of animals should be avoided if there is unnecessary duplication of pertinent studies. For this reason, we, biomedical engineers, should first and foremost do our, what I will call, homework. Namely, if and when our project reaches the point of deciding whether to conduct animal experiments, we must check very well and see whether similar experiments were conducted in the laboratories or companies where we work, and in addition, whether such experiments were conducted by other research laboratories and the results have been reported in the literature. Such a review will avoid the unnecessary duplication of experiments and, of course, will not use unnecessarily animals in small or large numbers. Concerns regarding the use of animals in research have promoted searching for alternatives. This approach is exemplified by the 3 R's: reduction, refinement, and replacement. An interesting current promising approach is the establishment of the Medical Device Development Tools Program in the Food and Drug Administration Center for Devices and Radiological Health. The purpose of this program is to establish and explore replacement for animal tests. Some novel approaches are, of course, in vitro tests. We mentioned and we reviewed the use of cultural cell models as one of the stages of the evaluation of the biocompatibility of materials and devices. Another very promising new technique is the so-called organ-on-chip models, which of course have put together laboratory in vitro systems which are very sophisticated and are working in such a way that they can expose samples of novel materials to not only one type of cell or to one type of fluid or condition which reflects the human body, but many types of cells, different body fluids, and to find out whether those conditions will promote biocompatibility results and what are the other requirements to bring about the formation of new tissue under those circumstances. This is a very new and very promising, extremely sophisticated, of course, approach and model. in addition to knowing the spatial aspects pertinent to research using animals, biomedical engineers who are considering using animals in research must also be aware of compliance requirements. Individual investigators, group of investigators, and institutions where such research is conducted must comply with all local, state, and federal laws and regulations regarding animal tests. We should keep in mind that the United States Animal Welfare Act of 1966 and subsequent amendments addressed various aspects regarding the care and use of laboratory animals, have established federal laws, and they have set a system for monitoring and controlling animal-related research. Compliance to all aforementioned regulations and laws is required by the federal agencies which fund research, for example, NIH, and compliance is scrutinized by review committees at the local institutions such as IACOX, which review and approve animal-related research in universities, medical schools, and industry where such research is conducted. In closing, animal tests are serious and major undertakings. They must be considered only when all the preceding types of tests are completed successfully. The animal experiments are expensive. Remember, they require special facilities. They require the procurement of animals and the maintenance on a daily basis. Don't forget, the animal facilities charge per diem, which means something like a rent and food and care of these animals. Properly trained personnel are used in these facilities, including, of course, veterinary doctors who maintain the conditions which are needed to keep the animals healthy. Animal experiments are complex. We are dealing now with the response of our favorite material and device into the complex body of animals. And as a result of that, the results of these tests are sometimes difficult to interpret. For sure, negative results make that particular material and device unacceptable to be considered further for the development of a specific product. Negative results seal the unacceptability of the tested material, device, and potential product. However, positive results do not necessarily prove or predict the biocompatibility of materials and devices in humans. One of the major criticism one received in this particular case is the following. Just because a particular set of experiments was successful in rabbits does not guarantee that this particular material and device will be successful in humans. The same way that, or in a similar way, the results which are successful when we do in vitro experiments do not always guarantee that that particular biomaterial and device will be successful once it is placed in the body of animals. All of these are very good arguments, but in order to complete the evaluation of the biocompatibility of implant materials and devices, these tests are needed in the order that we described, starting with in vitro experiments, going, if they're successfully completed, continuing with animal tests, and if those are successfully completed, moving and addressing clinical trials. Animal experiments are useful and unavoidable preceding clinical trials. Clinical trials, of course, are those in which the material and device is now used in humans. This aspect has raised a lot of philosophical discussions among people who do not believe or they are not supporters of animal experiments. Of course, this is a decision of the conscience of each one of us, but it is not appropriate. Actually, it is very, absolutely inappropriate and against all suggestions and all opinions to take a particular product and use it directly in humans without doing all the preceding tests and experiments we described so far in our review of the evaluation of the biocompatibility process of implantable materials and devices, including the animal tests. This is the end of Lecture 28, Evaluation of Biocompatibility, Animal Tests.